Thanks, Patty. I'd like to acknowledge that today that we're meeting on Larrakia land and pay our respects to elders past, present and emerging. We've been working on malaria pathophysiology for over 30 years now, and we think that the latest splenic findings are amongst the most interesting and exciting that we've found. Can you hear us? We're going to hear today from Steven Kho about the hidden splenic reservoirs of Plasmodium vivax and falciparum, and it's really changed the paradigm for the malaria life cycle in humans. And this hidden biomass has got real implications for malaria elimination, given that a lot of these patients were PCR negative in their peripheral blood. There's been lots of spin-off important findings with understanding the mechanism of loss of uninfected red cells as a major contributor to malarial anemia. And Stephen's going to be presenting how this is leading to a whole lot of exciting research directions, including with the support of his Ideas grant this year and the Ian Potter Foundation laboratory equipment support. So this was, as you know, Steven started this study as part of his, it's a high risk component of his PhD. He's really led this to amazing levels. So, Steven, thank you for telling us more about where the research is heading.
Great, thanks, Nick. Put this here. Okay, so firstly, thanks very much, everyone, for spending time to come over to my seminar. Basically, I'm going to talk about malaria and spleens. And this is a modified version of a talk I just gave last month at an international malaria conference in Barcelona. And it comprises both published and unpublished work from ongoing projects within the malaria spleen program that Menzies leads, in collaboration with many international partners. And you can see here some of the institutes involved at the bottom, based predominantly in Indonesia and in France. So I'm really giving this talk on behalf of a large group of researchers and collaborators that have been involved in the work. So firstly, just a quick slide on malaria. It's a parasitic disease caused by protozoans of the genus Plasmodium. It's transmitted by female Anopheles mosquitoes, and there's 7 species known to infect humans, with falciparum and vivax being the most common. In 2021, the World Health Organization reported close to 250 million cases of malaria and just over 600,000 deaths worldwide. And what's really worrying is that these numbers have increased over the pandemic and have been relatively static in the years before that. And as you can see in this incidence map, most malaria cases are concentrated in Africa, followed by Asia and South America, and with the majority of deaths occurring in children under the age of 5. So malaria is really still a significant health problem today. This is a summary of the parasite's life cycle that we've recently generated based on the latest evidence in humans, with falciparum on the left and vivax on the right. Oh. Sorry, how do I? Yeah. So I'm not going to go into this in a lot of detail, but essentially within the human host, sporozoites from the mosquito enter a liver stage of 7 to 14 days before they are released as merozoites into the bloodstream, where they infect red blood cells and form this intravascular erythrocytic life cycle. We also know of the bone marrow reservoir, responsible for development of sexual stages of the parasite, which eventually return to the mosquito to enable ongoing transmission. And our contribution to this illustration, based on these two recent papers that we published not so long ago, concerns the presence of a large reservoir hidden in the spleen and the existence of novel intrasplenic life cycles for both species. So this is the outline of my talk, where I'll first recap on what we did to uncover these findings. I'll talk about evidence we have supporting the endosplenic life cycle, firstly in vivax, and talk about this also in falciparum. And I'll end with future directions of our work. So how are we able to get our hands on human spleens? We've established a really unique splenectomy cohort in Timika, and this has really been central to all the human splenic studies we're running. Timika itself, it's an endemic area in Papua, in eastern Indonesia, with a high transmission of vivax and falciparum. We prospectively enrolled any patient undergoing splenectomy or spleen removal at the local district hospital. And what you see here are details of our first cohort of patients. We were able to enroll 22. And none of them showed malaria symptoms at the time of surgery, and the majority were also untreated, with no recent history of antimalarial treatment. Nearly all splenectomies occurred due to trauma-related splenic injury, mostly from traffic accidents. And 2 of these 22 were splenectomized due to elective surgery because of massively enlarged spleens. In terms of samples, we were able to collect entire spleens and also matching peripheral blood from the same patient. And we conducted a long list of experiments on site in Indonesia, with a minority also done overseas. So in 2019, we commenced recruitment of a second cohort of patients, and this is still ongoing. And now we have enrolled, I think, 47 patients in the second cohort. However, most of the data I'm showing today comes from the first cohort of 22 patients shown here. So these are some pictures of some of the spleens we collected in the first cohort. And as expected, we received spleens of varying sizes, most of which were enlarged. And this is a common manifestation seen in malaria endemic areas, the enlargement of spleens. So in the 22 patients from cohort 1, we first wanted to know who was infected with malaria parasites and where. So we used several different methods to detect infection, including rapid diagnostic tests. We also did microscopy of blood and spleen tissue, and we also did PCR. And as you can see here, quite surprisingly, we found that nearly everyone had asymptomatic Plasmodium infection. We had 13 with falciparum and 7 with vivax, and 1 mixed infection there in the middle. And what I like to point out on this slide is that if you think about the gold standard screening of peripheral blood by microscopy, which is what is used predominantly in the field and in hospitals, you'll be missing nearly half of the PF infections and all but 1 vivax infection. And even testing the peripheral blood by PCR, we were still unable to detect some of these splenic infections. And so this itself raises sensitive questions around current test and treat approaches that are being used for malaria elimination in some countries, as well as the accuracy of previous estimations of prevalence in endemic areas that only screen blood. So to explore parasite biomass in the spleen, we directly counted the parasites on Giemsa stain sections. And what you see here are snapshots of some of the sections in our cohort. And just to really emphasize, what we counted were these intact non-phagocytosed parasites inside red blood cells, and not those shown by these green arrowheads here, which are likely splenic macrophages that have engulfed parasites or parasite pigment. We also performed a number of microscopy validations, but probably the most important validation was the artificial perfusion of lab parasites into normal human spleens, shown here on the left. And this was really important because it allowed us to familiarize what each parasite stage looked like in this type of tissue and sample type. And it really served as a reference for our microscopy. So this is an important slide. This basically shows our key findings from the Timica matched spleen blood samples. And so using the microscopy data, we found that for both vivax and falciparum infections, the parasite densities were way higher in the spleen compared to matching peripheral blood, suggesting some accumulation. And so to explore differences between the two species that we examined, we simply calculated the ratio of spleen to peripheral blood parasite density. As you can see here on the right, the density of vivax parasites in particular was several thousand-fold higher than in blood, in the spleen than in blood. And this was significantly higher than the ratio in PF of around 300. So overall, this data supports the existence of a hidden splenic compartment in chronic infections by both of these species, with the extent of accumulation much greater in vivax infections. So next, if we focus on vivax, we calculated the tissue distribution of vivax biomass in our cohort. And we also estimated what the biomass might be in bone marrow using published assumptions. So these estimates indicated that the spleen makes up more than 98% of total body vivax biomass, while peripheral blood only accounted for around 1%, and bone marrow even less. So this is another really key finding from our study, and highlights that circulating parasites are only a small portion of total vivax biomass in the body. So this is chronically infected patients, but who make up the majority of global malaria burden. But what about in clinically ill patients with vivax malaria? So we've also recently been able to estimate this pie chart for clinical patients using PVLDH-based data from spleen intact versus splenectomized patients experiencing an acute attack. And as you can see here, while there's still a predominant presence in the spleen, a greater proportion of parasites are circulating and causing disease, which is what we hypothesize. So next, if we focus our efforts on understanding how this hidden splenic biomass might be formed in chronic vivax infections, we asked the question, could there be a cryptic vivax life cycle occurring inside the spleen? This brings us to the next part of my talk, where I'll present some of our evidence supporting the endosplenic vivax life cycle. So we first did some calculations to determine if the magnitude of vivax in the spleen could arise solely from retention of peripherally produced parasites. And in summary, we based our calculations on observed data from our cohort, as well as some published assumptions from the literature. And essentially the answer to this was no, where even after we incorporated all these conservative assumptions against a splenic life cycle, our calculations of intrasplenic vivax parasitemia of 0.13%, you can see there, could not reach the level that we observed in vivo, which was 0.8%. So one explanation for this difference could be the presence of an endosplenic life cycle. Secondly, we determined if all vivax developmental stages that make up asexual life cycle are present in the spleen. And as expected, this was the case. And also, you can see here a few examples of the different stages. And also the frequencies of each of these stages in the infected spleens were consistent with their duration in the 48-hour vivax life cycle from 2 permanent ex vivo studies. So thirdly, we explored if the spleen was a reservoir for reticulocytes, which are red blood cell precursors, and particularly the younger forms that express a marker called CD71. And so these cells are the known invasion targets of vivax. So to address this question, we phenotyped spleen and blood samples by flow cytometry. And as you can clearly see here in the middle, that the spleen was significantly more concentrated with these highly immature CD71 intermediate and high expressing reticulocytes compared to matching peripheral blood samples. We also calculated immature retics directly on CD71 stained spleen sections. And again, you can see here on the right, we found there was a major accumulation in the spleen. And so overall, the spleen does contain an enriched pool of cells that serve as invasion targets for vivax life cycle. Fourthly, for parasite replication events to take place, the parasites and the invasion targets need to be colocalized in the same area. And of course, this was the case in our infected spleens, whereby we found that 99% of vivax parasites and 92% of reticulocytes were colocalized together in the splenic red pulp. And as you can imagine, the red pulp, it's the perfect environment for successful invasion, reinvasion events to take place because of its reduced blood flow and shear stress compared to what happens in circulating blood. I also wanted to share some pilot work which was funded by a Menzies small grant last year, where we explored if chronic vivax infections induces red cell production in the human spleen as an additional source of reticulocytes for intrasplenic replication. As you know, this process of red cell production is not normally seen in the spleen in humans, and it's physiologically restricted to the bone marrow. So to start to explore this, we've screened spleens in our first cohort of 22 patients for clusters of nucleated cells expressing the erythroid marker glycophorin A or CD235A, as well as CD71. So nucleated cells that co-express these markers are erythroblasts, and erythroblasts are the very early precursors to red blood cells. And when present in clusters, as you can see here in some example images, these can be considered as erythroblastic islands, and these are strong evidence for red cell production. So at the moment, we are only able to do this using serial sections, but we very interestingly found clusters of these cells present in the red pulp in 2 vivax infected spleens. An example is shown here, where on the left panel, you can see a cluster of nucleated cells expressing glycophorin A. And this is depicted by the brown coloration. The brown coloration is showing positive signal for that marker. And in the same exact area on the right, on the serial slide, expression of CD71 on these cells. So it's really strong evidence that these are erythroblastic islands. So to give you an idea of their frequency in the red pulp, this is a snapshot at very low magnification from one of the vivax infected spleens. Hopefully you can see these very dark clusters of these cells, which were relatively common throughout the architecture, but mostly, or all of them were in the red pulp. And so to convince us even more that what we're seeing were erythroblasts, we recently examined CD71 based magnetically enriched spleen samples, which we stain with new methylene blue. And we observed these stages of stage 0 nucleated cells, which based on Hallmeyer's classification of retics on the left here, these correspond to late stage erythroblasts. We've now also started flow cytometry experiments to understand the different stages of erythroblasts in the splenic homogenates based on published markers. And shown on the bottom right here is an example from patient 4 who happened to have mostly late stage orthochromatic erythroblasts in the spleen. So what's next? We're going to screen more spleens from our second cohort of 47 patients, and we'll then do some formal quantitations of these erythroblast clusters, really to nail down that what we're seeing is evidence of red cell production. And we'll also look at this in infection by falciparum, whether they're present there or not. So to put this all into context, we've proposed a model of how the endosplenic life cycle might be taking place. In summary, we think that the majority of invasion reinvasion events take place in the slow-moving chords of the splenic red pulp, and this generates the majority of the splenic biomass. However, there's also potential replication events taking place in the fast circulating zones of the spleen due to a minor presence of asexual stages and reticulocytes in these areas. And the parasites produced here could potentially serve as those that are destined to exit rapidly from the spleen, such as the transmission stages. So what about in falciparum? Well, this is still a work in progress. We think there's also an endosplenic life cycle, but probably not to the same extent as vivax. So what evidence do we have so far? Firstly, similar to vivax, we observe all asexual stages of falciparum in infected spleens. And these were predominantly in the cords of the red pulp, as shown on the right, which, as I said, is most likely the area where successful invasion reinvasion events take place. Other evidence supporting this was simply the ability of the parasites to grow parasites from the spleen. We were able to grow them in culture, meaning they were viable. And as shown here, we were able to grow the splenic isolates over several cycles. We also did some similar calculations as we did with PV to determine if the magnitude of falciparum in the spleen could arise solely from peripherally produced parasites. And again, even after incorporating all these conservative assumptions against a splenic life cycle, we could never reach the numbers that we observed in vivo. So this could probably only be explained by the presence of an endosplenic life cycle in falciparum infections. Another interesting observation in our Timica cohort was that the spleens were enlarged in falciparum infections. And so when we tried to understand this at a macroscopic level by examining the architecture of spleens of varying sizes, we found that there was expansion of red pulp in larger spleens, paralleled by a relative loss of white pulp. And when we look at it more closely in the red pulp, we could clearly see that this expansion was driven by congestion of mostly uninfected red blood cells. And as Nick said, it also happens to be that this congestion is a cause of the majority of anemia that we see in malaria. So what does this mean for the parasite? Well, to put it simply, the congestion of red blood cells, especially in falciparum infected spleens, means there's more target cells for the parasite to invade. And if you look at this mature stage here on the right, it's surrounded by red blood cells. And you can imagine when this Schizon ruptures, those merozoites will immediately invade the surrounding red blood cells that are almost static. So the last thing I want to share are some results from electron microscopy work we've been doing looking at knobs. And so these knobs are a congregation of proteins on the surface of mature stages of falciparum that cause them to be sticky and allows them to sequester. And so it's been hypothesized in literature more than 10 years ago that if an endosplenic falciparum life cycle exists, mature forms are likely to be knobless. So this is exactly what we explored. Shown here are some positive controls where we perfused lab strain PF falciparum into a normal spleen, which hopefully you can see on the screen, they're very knobby. And on the other hand, when we looked at Timica spleens with natural chronic infections, mature falciparum stages were almost all knobless. And shown here are some examples which were generated in Jakarta from at the Eichmann Institute. So what does this absence of knobs mean? Well, they probably have reduced cytoadherence capacity. They're less sticky, meaning they're probably accumulating in the spleen by biomechanical trapping. You can also then start to speculate about how this may contribute to their survival in the spleen, including immune evasion, energy conservation, and reduced virulence. So this is my second last slide and shows the future direction of the spleen program. We've now have a total of 69 spleens from the 2 cohorts, and we're branching out into different disciplines. Some of the work which I've already talked about and others that I have not. In terms of diagnostics, we're currently testing the detection of hidden splenic infections using a panel of serological exposure markers developed by Ria Longley in Melbourne. One of the most exciting avenues is parasite transcriptomics to understand the pathobiology of the splenic population and whether they behave differently to matching blood isolates from peripheral blood. And we're also interested in elucidating the genetic diversity of the splenic population of parasites compared to what's circulating. And all these proposed genetic and transcriptomic studies were recently funded through my NHMRC Ideas grant. And we've also recently secured funding to purchase a new cell sorter for the Menzies lab. And this will facilitate the generation of high quality samples for these studies. In the modeling space, very briefly, we recently submitted some work for publication that modeled the dynamics of Plasmodium vivax lactate dehydrogenase. And this is a parasite biomarker that is present in blood and could potentially be used to estimate hidden parasite biomass outside the bloodstream. In the immunology space, we're collaborating with Michelle Boyle at the Burnett Institute to better understand malaria immunity and how the parasite and immune cells can coexist in the splenic environment. And we're also starting some spleen cohorts in Africa to study questions such as the hidden dry season reservoir of falciparum. So finally, I wanted to leave you with a key take-home message. I hope I've convinced you based on our findings that at least in chronic infections, malaria should be considered predominantly an infection of the spleen. And with that, I'll just like to end there and thank everybody who's been involved with this work. Special thanks to my mentor, Nick, to the wonderful teams in Timika, and to our collaborators in Jakarta and Paris, and of course to all our funders. Happy to take any questions if there's time, sorry.